So Raka, how are you doing in your 126 class?
Oh,
126 class?
my students are fantastic. I love them.
Oh, I think I do too. We're having a wonderful Bio 125 class. We're going to have some sort of little competition at
Oh,
the end
hey
of
Stephan,
Bio
Stephan,
125,
I have a burning
126
question
semester.
for you. It's about the structure of DNA.
I got a few minutes. Raka, I'll see you later.
See you
We'll
later.
talk about the course.
Raka.
Let's come into my office.
All right, this is a big one.
So what was your burning question that you had from reading the textbook there, Mark?
Well, so I'm reading the textbook in detail, like we do, and I'm looking at this phylogeny of bacteria. And right here on the page, it says a whole phylum of bacteria is low GC, and another phylum is high GC. But right in your lecture, you talked about Chargaff's elegant rules about the structure of DNA and how A and G and T and C have to have these exact ratios. How can these organisms have DNA and have different amounts of G and C? Doesn't that break Chargaff's rules?
Well, Mark, I think you misinterpreted Chargaff's rule, and I just so happen to have Chargaff's rule on my whiteboard here and a couple of his data points. But remember from class that Chargaff's rule states that for any given organism, for any given organism, the percentage of A's will equal the percentage of T's, and the percentage of C's will equal the percentage of G's. And these are some actual results that Chargaff did as he broke open the cells and asked the percentage of A's and T's and C's and G's. In a human, for instance, 60% of the nucleotides are A's and T's, 40% are G's and C's. And you'll see it does vary, though. The rat is 56% A's and T's, yeast at 64% A's and T's. So within a human, 30% are A's, 30% are T's, 20% are G's, 20% are C's. It doesn't have to be uniform, and in fact, the fact that it differs between species actually lent some credibility that DNA is genetic information, that there is a bit of difference between different species.
Well, that makes sense. So it could vary, but why would it ever vary? Why would it go between different amounts of G and C or A and T? Is that just random?
Well, there is a bit of randomness in it, yes, but I like the example you brought up, these bacteria, and if you look closely, those high GC content bacteria, those tend to be the thermophiles that you lectured on a little while ago. These heat-loving fellows right here, if you look at their GC content, it's almost sort of off the chart. It can be greater than 60% of the nucleotides are G's and C's. Now, why would an organism have an evolutionary adaptation to have a greater amount of G's and C's? Well, if you also remember from lecture, between A and T, between adenine and thymine, that base pair has 2 hydrogen bonds, whereas a cytosine and a guanine has 3 hydrogen bonds. A considerably stronger non-covalent interaction than the non-covalent interaction between A's and T's. So the theory is the evolutionary adaptation to living in a very hot environment to prevent the DNA from separating is to have a higher GC content than other organisms who aren't faced with that environmental constraint.
That makes total sense. Chargaff is saved.
Chargaff is saved.
Thanks, Stephan.